All right, so we're getting into Mendelian inheritance. Last time we talked about mitosis and we talked about meiosis. Mendelian inheritance is really just the application of meiosis. Now, what happened during meiosis? Do you guys remember? That's right. So the cell, the single diploid cell, 2n, is going to split into 4 haploid cells, n. And those haploid cells were genetically unique because what happens during meiosis? What's a big event that happens during meiosis where you get mixing up of stuff? Okay, let's start at the beginning. During the beginning part of meiosis, you get the chromosomes sort of lining up with each other, the homologous pairs. What's a homologous pair? They're the same chromosomes. So they've got the same, they're encoding for the same genetic material. So they're going to line up, but instead of them being separate pairs, they're going to come together. And they're going to form what are known as tetrads or synapsids. I don't care.
2 pairs coming together.
Right, 2 pairs coming together. Now they're really, really close to each other, and they're genetically very similar. So at that point, they can swap parts. What's that swapping called? They form an X shape, and then one leg and the other leg switch places. Actually, that's really close to translocation. That's a great idea, but it's wrong. But it's really, really, really close. No, you got that's the absolute right concept. I totally get why you said that. No, it's crossing over. There's a crossing. They flip. So you're able to move genetic material from the paternal chromosome to the maternal chromosome, and the maternal chromosome to the paternal chromosome. So these tetrads line up together. When they've lined up along during metaphase, you're going to have the microtubules connect to them, and they get pulled apart to either end of the cell. Recall this? That's right, during anaphase. Now, in anaphase, you could have a few things happening. This is where you're going to get independent assortment and segregation. So, let's see, how can we do this the easiest way? Can I use your fingers real quick?
Oh, wow.
Yeah, okay, I need 2 hands. Okay, so make chromosomes like this. Yeah, good. All right, so we got, hold them up. Good. Our fingers are different. You can tell the difference, right? Yeah, good. Okay. So, tell you what. Let's go with, yeah, the stick game. Okay, so this one hold up this finger, good. This one hold up your pinky. Good. All right, so they've lined up like this, right?
You have small fingers.
You're a lot bigger than I am. All right? So what will happen is they're going to replicate. Now, it's probably better to do it like this. Sorry, back to this. So, homologous pair, they've replicated. They've formed a tetrad. Bring them all together. All right, awesome. They're all together. Now, into the daughter cells, we could go, pull them toward you, could go like this. You can be the dad. You're paternal, I'll have the maternal ones. Or it could go this way, at which point you have 1 and 1, 1 and 1. Totally random, entirely random. It's that part of meiosis that ends up forming the basis of Mendelian inheritance. Because it's 50-50 shot. All right, so that's really, really useful for us in the classroom. So whoever's watching this on video, that will make no sense to. So at the end of meiosis, or beginning of meiosis, you've got a diploid cell with paternal and maternal chromosomes. Crossing over occurs, so you end up with unique recombinant chromosomes. By the end of meiosis 2, you have haploid cells that are all unique. Those then will be able to bind with each other. A fertilization event can occur. Now, the big thing to remember, huge takeaway, this is an entirely random distribution. Whether you get your mom's chromosome or your dad's chromosome in any given gamete. Totally random. 50-50 shot per chromosome. Yes?
Is it really 50-50?
totally 50-50 shot.
It's like something they have like a lot of girls. It seems like the only thing the guys spit is like girls.
Yeah. But it's just like if you flip a coin 20 times, you got a chance at hitting 20 tails. There are, I will say, and it's a little bit more advanced, we'll talk about it more when we get to the genetics of sex chromosomes, some genes that will make a male inviable, which means if it's on the Y chromosome, the male is dead. I'm sorry, or if it's on the X chromosome and it's an X chromosome, I'm getting way ahead of myself. But surprisingly enough, we figured out a way to control mosquitoes using this. Yeah, they kill off all the males. Yeah, females spread the disease. Because really, there's no point to a mosquito. Working backwards with the chromosomes. Remember, when in meiosis, we talked a lot about chromosomes and chromosome movement. We have to remember that chromosomes are made up of DNA, and DNA is genetic material. DNA are your genes, the instructions for building you. So when we're looking at this piece from your mom or this piece from your dad, this chromosome from mom, this chromosome from dad, those are the instructions that are necessary to build you. What is a polypeptide encoding region of a chromosome called? Something that will encode for a certain protein. We've mentioned it a few times in class. What is it that actually, it's made up of DNA, and it's the plan to build you? It's located in the nucleus. This particular bit of information is then transcribed to make RNA. Specifically, you're right. Think a little bit bigger picture. No. Okay. We have talked about genes in here, haven't we? Yeah. Okay. A gene is a polypeptide encoding region of DNA. I'll be dead honest. It was probably at this point in the semester, I was teaching a genetics class, a 200-level genetics class, so much harder than this class. And I asked the students that same question, what's a polypeptide encoding region of DNA called? And they stared at me blankly for 10 minutes. And I got so mad at them that I just kicked them out of class. I was like, we're done. I can't teach. This is literally what the whole course is on, and you don't know. So if an alternate form of a gene is an allele. And that's what we're really getting into today. How are alleles inherited? No, it's cool. So genes are polypeptide encoding regions of DNA. So on the chromosome, you've got long strands of DNA. A small region of that is going to encode for a specific gene that will make a specific protein. You guys are looking kind of lost on that idea. Let me draw it out. That might help. Gene. Yes? What's a gene pool? Next semester. First lecture, gene pools. We'll talk about it. Basically, a gene pool is when you're looking at a population level gene structure. So not an individual, but the whole group. All right, so here we've got DNA. All right, DNA. DNA is going to be composed of a whole bunch of monomers. What's the monomer of DNA? The nucleotide. And there are 4 of them. Guanine, adenine, thymine, cytosine. And I'm just going to put a whole bunch more just for fun. So you got a gene. That's going to encode for a bit of RNA. So this is DNA. That DNA is going to encode for RNA. So that specific strand of DNA, that could be really long, will only encode for a shorter strand of RNA, because that's the only part that's the gene, this part. That's then going to be converted into a polypeptide. So polypeptides made up of a bunch of amino acids. I don't know, tyrosine and lysine. So you have a small piece of the DNA is going to be converted to RNA, and that RNA is going to be converted to an amino acid sequence. Get this then. This is the part you guys already know. The amino acid sequence is going to be super long, right? One big long chain called the primary structure. The primary structure starts to fold up on itself to create secondary structures. They're going to go on to make tertiary structures, and the tertiary structures work together to form a quaternary structure that gives the protein a form, which gives it a function. It all comes back to this, which is called the central dogma of molecular biology. So, inevitably, you guys, I've told you several times, the whole final exam is about proteins, right? It's because really, it all deals with proteins, top to finish, start to bottom. That's totally backwards. Sorry about that. Reverse it. Do it again. Is the cell cycle included in the test? Yes? Sorry, that was a little off, but we're good. We're good. So we're going to be making these genes. We're looking at these genes and seeing how they segregate, how they sort. Because that DNA is found on those chromosomes. So, I'm sorry, that DNA makes those chromosomes. Really, a chromosome is just really tightly wound up DNA. All right, so this is what we know and how we know this works. But a long time ago, they had no idea about this. Long before we knew about the structure of DNA, long before we knew that DNA existed, we knew about inheritance. And there's this guy named Gregor Mendel. He was a monk in Moravia, and he was an avid pea breeder. Now, you can imagine that he was the life of the party. I mean, he's a mathematician, he grows peas, he's a monk. Awesome. What? He can't. No, some monks can talk. It's only if you take a vow of science. But he could write profusely, too. He was the first person who really looked at breeding with a really, really serious mathematical approach. Before Mendel, people bred stuff. I mean, that's sort of like what they did. But he was the first one who wrote it all down and did some math. Now, that's the big difference between science and just screwing around. Writing it down. You guys have all done really stupid, dangerous stuff in your life. Usually, you know, when somebody dared you, or you thought it was a good idea and it would make life easier and it really wouldn't. You've done stupid things, but did you write it down? Because if you wrote it down and then replicated it, then it's science. Just saying. Next time, write it down. Then somebody pulls you over. Somebody like a police officer. What are you doing? Science. I did this 3 times before. I'm doing it again. It's replication. No, you know what? It depends. It really does. I don't know the police in Chesterfield right now. You're not? Say that again? You got a warning? That's nice. I got pulled over an awful lot when I was in the southern part of the state because I looked a little different than everybody else. Yeah, and so I don't know what the police are like up here, but I'm really glad to not be there anymore. They're like, okay. Then just don't cross the line. All right, so in this lecture, I've interspersed slides like this because I'm going to start using big words, and I need you to know what they mean. Okay? Hybridization, traits, self-fertilization, true breeding, and cross-fertilization. When you get to these slides, I need you to know these words because if you don't know these words, you're not going to know the questions I'm asking you on your test. And the questions aren't going to be, what are traits? The question will be something along the lines of, no, not even describe traits. Let's say, I don't know, self-fertilization. A pea plant self-fertilizes, a tall pea plant self-fertilizes. Give me the F1 ratio of its offspring. Exactly. That's why it's important to write these words down and know what they mean. Don't worry, we're going to go over them in the next several slides. Good to go? Yep.
I wanted to jump in here months after I originally recorded this to fix this slide. I want to tell you that a trait is a variant of a character. So if you think about it, the character is the broad category, and the trait is the possibilities of that particular character. So character is general, trait is specific. In this case, if we're looking at the trait Widow's Peak at the very top of the screen, that is, I guess the character would be hairline, and the trait would be Widow's Peak or not Widow's Peak. In the middle picture, you have polydactyly, somebody with 6 fingers, which, as you know, is done with AI or apparently with actual genes. Most people get this a 6-finger removed. It's a dominant trait, but the character would be finger number. The trait would be 5 fingers or 6 fingers. The ability to roll the tongue is also genetically determined. So ability to roll tongue would be character. Tongue roller, non-tongue roller would be the trait. Mendel looked at many different characters. I think he looked at 7 different characters. He may have looked at more, but just really focused on these. And each of those characters had 2 traits, and only 2 traits. So if we're looking at these flowers here, we have flower color is the character, and white color or purple color are the traits. Down here we have seed shape. Seed shape could be wrinkled or it could be round. We also have seed color here. Seed color is the character. The traits are yellow or green.
All right, so he was doing all these breeding experiments because you can't really breed well with people. Or they don't like it when you try to watch. Yeah, it's like... So what he was doing at that point, what everyone was doing, was what's called self-fertilization. Self-fertilization is how we created the plants that we have today. We would take a female gamete and a male gamete from the same plant, put them together, and you end up with offspring that look remarkably similar to the parents. If I took a pollen from a red flower and an egg from a red flower, and I put them together, what am I likely to have? Red flowers. You do this over and over and over again, and you get what's called true breeding. The same alleles over and over and over again. So the same genes again and again and again and again. This is a lot like how we get modern seeds and plants. You guys have ever gone out and bought seeds to plant in a garden before? Okay, now here's the fun part, right? You look at the package, and you see what that plant's supposed to look like. And then you plant it, and you expect that plant to look like that picture, don't you? That's because those seeds are bred using true breeding. Time and time and time again, the same traits are crossed, the same traits and the same characters of that trait are crossed to each other. So this eventually results in the same trait generation after generation. That's called true breeding. We have done this forever. We've done this with corn. We've done this with wheat. We've done this with horses. Heck, we've done this with dogs. We've just, about 150-ish years ago, we were like, you know what? We want cool-looking dogs. We want the most messed-up-looking dog you can possibly come up with. And that's where we created dog breeds. Like your dog, yeah. What? Yeah, there's no point to a bulldog wouldn't survive in nature. There's no point.
Like German Shepherds are not supposed to have this bent back legs either.
Right. But people decided, we like this trait because it looks cool, and I wanted this dog with hip dysplasia and a squished nose or whatever. No, it sounds bad. Truthfully, a lot of true breeding doesn't work out terribly well for the animal. Friend of mine, I don't know why, she had to have this like breed of cat, and this cat has a really squished flat face, to the point where it's really hard for it to eat and breathe at the same time. And it, I guess, I don't know what word it would be. It's like snorkel, I guess you'd call it, when it tries to breathe. It's like, ah. I hate that thing. But then this cat also has like severely depressed and is on antidepressants and truthfully, yeah, your dog's on anti-anxiety medication?
Yes. It's my dog.
What kind of dog is it?
He's a Shorkie.
Is it Shorkie? Is it a purebred or close to? Your dog's a mutt? Your dog's just messed up because it's living with you. Oh. It's an angry dog. Everybody knew about true breeding. Everybody knew if you cross the same, you get the same. You take these dogs that look exactly alike, and they produce puppies that look exactly alike. But what Mendel was interested in was what's called cross-fertilization, where you take the pollen from one flower and put it on the stigma of another flower on a different plant. He wanted to take two individuals with different characters of the same trait and cross them together. That's called hybridization, where you take two different characters for the same trait and cross them together. And that's where things start getting wacky.
So 2 different characteristics
Of the same trait
and then put them together.
and put them together. So, on paper, he studied 7 traits, each with 2 characters. He studied the form of the seeds and the color of the cotyledon, the color of the flowers, the form of the pods, the color of the pods, the placement of the flowers, and the size of the plant. So there were either it was a gray and round seed or white and wrinkled. Cotyledons are, let's see, have you had edamame before? All right. So when
The seed inside.
yes, inside the seed, they'll have either a single cotyledon or 2 cotyledons, which is sort of like, you know, when you eat a peanut, how it splits,
Yeah.
like that. They're filled with sugars and fats and everything that a baby plant needs to survive. Again, 102 topic. We'll get into cotyledons. It'll be awesome. It is. It's really exciting. We're going to talk about how babies grow, plants, and real babies, but they're not nearly as interesting as plants. Flower color, white or purple. The form could be full, which means really thick, or constricted, which sort of you can see lots and lots of bumps in it. It looks like edamame. The color could be yellow or green. The placement is what's termed axial, which means off to the sides, or terminal, which is up at the tip, or the size is either long or short. So we studied those 7 traits, each with 2 characters to them. All right, more terminology. Monohybrid, P generation, F1 generation, F2 generation, dominant recessive genes and alleles.
So genes and alleles are different.
Alleles are alternate forms of genes. So first off, monohybrid cross. This is where you're going to cross a plant with one character of a trait with a plant with another character of that same trait. So they're variants of the same trait. Here we have a tall crossed to a short. We are only looking at one trait, thus monohybrid, mono meaning one. In this monohybrid cross, you're going to start off with 2 plants. Those first 2 plants you start with, the mom and the dad, are going to be the P generation, or the parental generation. They're going to produce a bunch of offspring, kids. That's called the F1, or the filius one, the kids. Then you could cross those kids together. Don't think about it in human terms, think about it in plant terms, because otherwise your brains will explode. Again, next semester, incest, we'll talk about it. That F1 generation crosses to itself. That produces the F2 generation, or the grandkids. So the parents mate, they produce the kids mate, they produce the grandkids. P to F1 to F2. It could. After F2, though, if you've got a hybrid cross, it gets a lot messier. So I'm never going to have you guys go beyond F2. In fact, I wouldn't even see you going beyond F2 in your undergraduate work with this. Yep. Down the line. What Mendel noticed was that if he was to take two pure breeding or two true breeding strains, that is, that they would always produce the same generation after generation, take two pure breeding strains of different characters for the same trait and cross them together, the entire F1 generation looked like one of the parents. So if he took a pure breeding tall and a pure breeding short plant, he crossed them together, every single one of the offspring would have been tall. No in-betweens. No mixes. I know. He didn't know. That's the whole thing. That's why this was really interesting. And that's the exact right question to ask. Why is this happening? He found that with height. He found it with seed shape. If he took a pure breeding round and a pure breeding wrinkled and crossed them together, he ended up with every single one of them as round. That's what's coming up. Yeah. The yellow and green cross them together. Every single one's yellow. Green and white seed coat, pod shape. You know, he kept finding the same thing over and over and over again. So the trait that he saw as always there, the one that was in the first generation, he termed that dominant. He termed the other one recessive. So the dominant trait is going to overwhelm the other. You guys know what the word dominant means? There's like a dominant personality in the room. Yeah, they're the strong one. They're the one that you always see. They're the one that is the loudest. They hide everybody else. The recessive one's kind of in the background. You guys know people who are dating, I assume. You might be dating someone. Nope. If you look at this couple, there's usually one person in that couple who's sort of the dominant one. They're the louder one. They're the one who's usually in control of what's going on. And the other person is sort of a little bit quiet and just goes along with it. It's that dominant one, that loud one, that gets their way. Make sense? It's cool because nobody argued with me this time. Usually somebody's like, no, we're in an equal relationship. And it's like, no, you're the recessive one. You're the recessive. I'm totally the recessive one. My wife gets her way all the... It's gotten to the point with me. Yesterday, my son wanted pizza and my daughter wanted Mexican food. And I said, well, I'm better with Mexican food. I think we'll do Mexican. And he went and he said, okay, I'm going to talk to Mom about this. I was like, okay. And so she talked to him and he said, Mom, you have to make the decision. And she said, well, your dad already said that's what he wanted. And he said, yeah, but Dad will do anything you say. So all you need to do is say you want tacos. And then he started singing a song called like Tacos Raining from the Sky. And he put it on YouTube for an hour and a half, the same song on loop. And that was the end for me. So we got tacos. Because that's what my wife said. There you go. So, let's see. Although I will say I was totally the dominant one in getting a door. I went out and my wife said, we don't need a door. And I was like, you know what? We need a door and I'm buying a door. So I bought a door. Well, so I can like stop water from going in my house. Well, I had a door, but it wasn't a good door. But now I got a good door. So, the visible trait, the one that you see in that F1 is the dominant one. The other one is recessive. It's being masked. In that F1, every single offspring was dominant. And that could make sense if there's some trait that will always dominate another. But it turns out in the F2 generation, so this is the grandkids, he took the kids, this tall kid and this tall kid, crossed them together. What they end up finding was, let's see, where's tall here? Here. 787 of the offspring were tall, and 277 were short. No in between. No mixes. Tall and short. That was a 2.84 to 1 ratio, about 3 to 1. He did this with all the other traits. 2.96 to 1, 3.01 to 1, 3.15 to 1, 3.14 to 1, 2.95 to 1, 2.82 to 1. All of these are pretty much the same. And that made him say, same thing you said, why? There was only one real explanation for this. The explanation was,
this mini lecture gave you a real sort of focus on the history of modern genetics. And we really focused in on Mendel, Gregor Mendel, who is traditionally known as the founder of modern genetics, the father of modern genetics. We talked in the beginning about sort of reaffirming what we knew about DNA, which is one of our 4 big macromolecules. And that on DNA, there are very special regions that are encoded by the molecules that can form polypeptides. And these regions are known as genes. So a polypeptide encoding region of DNA is known as a gene. Then we went back in time and we looked at what Mendel was thinking. Mendel came from a time before DNA, before we understood what chromosomes were. He was just looking at peas, and he looked at them with a very serious mathematical mind. He looked at traits, which are just variants of any particular character. He noticed that several of the traits were passed on to the offspring, but they were passed on in a preferential fashion. That is, some traits of 2 pure breeding parents would not be seen in the offspring, but then they would be seen in the grandchildren. So he started to do these simple monohybrid crosses, where you crossed one variant of a character, to another variant of the character. So one trait of a character to another trait of the same character. And that was known as a monohybrid cross, because you were just looking at one character. Mendel saw that in 7 characters in peas, I think it was flower color, flower position, seed color, seed shape, pod color, and pod shape, there were no blends of the parent traits in the offspring. It was either one parent or the other. The offspring with one trait with that was seen in the offspring, that trait was termed dominant, and then that trait that got hidden was termed recessive. It was masked by the dominant trait. So Mendel saw a lot of things, he did a lot of math, and he came up with the idea that there are 2 units of inheritance. And it was only decades later that we realized those 2 units of inheritance were the chromosomes in the cell. These content review questions are here to focus your study. In the next set of lectures, we're going to look at practical applications of Mendel's work. Specifically, we'll talk about how meiosis and chromosomes are involved in being able to predict what Mendel was able to find just using math.